project that I got involved in Dr. Duran's lab last summer. So starting with just a little bit of background, Chagas is a disease caused by the parasite Trypanosoma cruzi, and it can cause acute skin inflammation and inflammation of many body tissues, especially heart and intestinal tract. And the main form of transmission of the disease is by the insect vector, because once they are infected with the parasite and they are feeding on us, they leave the contaminated feces. And once we get in contact with this feces, we get contaminated with the parasite. And why we need to care about Chagas disease? It's estimated that 8 million people are infected, and a huge part of this number is in Latin America. And there is treatment available for the disease, but it shows many bad side effects, so many people can even not tolerate it. And in addition, there is no vaccine available, so the main form to control the cases is by vector control. And these vectors are also known as kissing bugs. And we have about 140 species of bugs, and here are some of the most important. So we see the presence of Rhodinus prolixus in Venezuela and Colombia in orange. And when we look to South America, we see the distribution of Triatom infestans. And as we see here in the map, Triatom infestans was to cover a huge part of Central America, highlighted in light purple, but this population was largely controlled by the use of pesticides. And when we look to Central America, north of Colombia and Ecuador, we see the large distribution of Triatoma immediata. And for these species, measures of control, like the use of pesticide, didn't work because this species has special characteristics that make this vector my objective of study. And the first of these characteristics is its large geographic distribution. So we can find Triatoma immediata from Mexico through all Central America, north of Colombia, Ecuador, and part of Peru. And the second one is the diversity of Triatoma immediata. We have diversity of morphology, as you can see here in the picture. We have bugs that are darker than others, like the ones from Lankin, others that are smaller, like the ones from Patton, and others that are bigger, like the ones from Quiche. And we also have diversity of habitats, so we can find Triatoma immediata in domestic, peridomestic, and sylvan habitats. And they also have different behavior, so they have good capacity of movement and colonization, and they also have different food sources, so they don't feed just in humus. Some prefer to feed only in humus, and others prefer to feed only in sylvan animals, so the ones that are in contact with refers to specimens from Yucatan, Mexico, Guatemala, and Belize. And the second one in red, called Timopana, that refers to specimens from a Belize cave. And we call these two clades that I just described as cryptic species because the bugs look like Triatoma immediata, but they are genetically different. And the third clade in gray scale is Triatoma immediata sensu strictum, that refers to specimens from other localities in Central America that are more closely related genetically. Acknowledgements to Dr. Dorn, my mentor, Dr. Carlota Monroy, our collaborator in Guatemala, Adrian Woods, our lab technician, and the program Science Without Burdens, and Dr. Tolman and her tropical ecology class for the book collection. Thank you.
caused by the parasite Trypanosoma cruzi, and it was first discovered by the Brazilian physician Carlos Chagas, and that's why it has its name in his honor. The disease was discovered in 1909, and it's the most serious parasitic disease in the Americas that can cause heart disease, which eventually can lead to death, plus the syndromes such as megaesophagus and megacolon. And by now, we have at least 24 mammal species that can carry the parasite. In the US, besides human cases, which are quite rare, Chagas disease has been reported and documented in dogs in Oklahoma, Texas, Louisiana, South Carolina, and there is an interesting case on Virginia where a female pack rat and 8 out of 9 of her puppies were found infected. So therefore, this disease is a zoonotic disease, which means that the disease or the infection are in the wild and can be naturally transmitted from mammals to humans. And as an example, dogs can be infected by the contamination of existing wounds, by ingestion of the bugs, or by transplacentally. Talking about a little about animal reservoirs. First of all, reservoirs are animals that can carry and keeps the disease in the wild. And the most common domestic reservoir are dogs. And to date, there is no cases of domestic cats infected. And in dogs, as you can see here on the top left picture, a dog with Chagas. And the symptoms are, as we heard in the previous presentations, symptoms are not as specified. So dogs can have loose weight loss, and if we do X-ray, we can see the cardiomegaly, which is heart disease. And as I said, there's no case of domestic cats reported. Talking about wild reservoirs, which is more related with my research, the first report we have of wild mammals infected was a pack rat or wood rat from California, and the state where we have most of our data is Texas, where 32% of pack rats were found infected, but also armadillos and opossums are also infected. This is a table that shows wild animal reservoirs that have been reported with the disease, and it's in chronological order. As we can see, the first case that was reported was a pack rat from California, and there is a lack of studies in pack rats from Arizona. And this is a geographical distribution of the previous table. So here we can see that the most common reservoirs are pack rats, armadillos, ringtail cat, and the ground squirrel. Raccoons, opossums, and mice are also important natural reservoirs of the disease. And as I said, there's no case of cats infected with the disease. And this guy over here, which is called the ringtail cat, is not a cat, it's actually from the raccoon family. Going into pack rats, which is my approach, these little mammals, they can live in a very, very diversity of habitats, from very cold icelands to dry deserts. They fed primarily in green vegetation, and a female pack rat can have from 5 to 8 offspring once a year. Our first infection that had been reported was in California, a pack rat from California. And as you can see in this map, the disease, pack rats, the prevalence of T. cruzi in pack rats are more relevant in western regions, for example, California, Arizona, New Mexico, and Texas. And in eastern regions, raccoons and opossums are the most common reservoirs of the disease. So with all this knowledge and lack of studies, I asked the main question that leads my project. What is the prevalence of Trypanosoma cruzi in pack rats from Arizona? To answer this question, we worked with 38 pack rats that Dr. Just Smith sent us from Arizona. We did dissection and DNA isolation from the heart tissue by Qiagen, and then we performed the electrophoresis gel. And in our population, we had 30 male pack rats and 80 female pack rats. And in my conclusion, first of all, why do I think this study is relevant? Because there is a lack of studies in pack rats from Arizona. So my acknowledgement goes to Loyola University, my mentor, Dr. Patricia Dorn, our partner in Arizona, Dr. Justin Smith, our research and lab technician, Adrian, and the Brazilian Scientific Mobility Program, Science Without Borders, that bring me here and gave me this opportunity. And also, just a thank you note to Rafaela and Lisa for teaching me every single day, and Barbara for listening to me like forever. Thanks.